the Neptune Project, in partnership with the National Science Foundation, the W.M. Keck Foundation, the University of Washington, the Pacific Northwest Gigapod, and the Research Channel, is proud to present Visions 05, Expedition to the Underwater Volcanoes of the Northeast Pacific.
We got a really serious problem here this morning. Yesterday many of you saw me standing on this deck with placid sea behind us, ready to do a program from the sea floor. Right now we got very serious weather. It's roughly 14 hours later, and the captain and the expedition leader are meeting me here, looking at the weather. We're going to decide whether to keep going, hold our position, or come out of the water. So that's what's happening here right now. Expedition leader is responsible for Jason and the safety of Jason and all the people who work Jason, and the captain is responsible for the security and the safety of the entire crew, everybody on board. And we need to make a decision at this point because the weather has gone foul in about 14 hours. So Phil, your feeling is that right now it's not tenable to continue. What's your view of what's going on?
We're first concerned with the safety of the ship and the people on board and the people that have to recover the vehicle. And then last of all, we're concerned with the safety of Jason, and it's getting to the point where we need to pull the vehicle.
Well, I concur, Phil. The problem we're going to face too is that the ship's DP system, dynamic positioning, is taxing itself right now in this weather. It's going to only get worse. That system is intensifying, as we all know. And so I think it's time to pull it.
So what's the recovery like? I mean, I agree with you. This is pretty serious. What's the recovery liable to be like? Is that something that is actually dangerous at this point?
We have to be careful. We've done it in these seas before, and we're not past the limit, but now's a good time to get out before it gets any worse.
And I was in the van with you less than 2 hours ago, and we were still working the sea floor.
Right.
Does this kind of thing, when you guys are in the water elsewhere, does this come up as well? This kind of, the weather goes bad, eh?
We always have to deal with the weather. Yeah.
All right. So
not the first time.
So there's a situation where the decision's being made to pull Jason out of the water, basically to protect Jason and the people that are involved with it. We simply can no longer work on the seafloor, and it's now getting dangerous to work on the deck to recover the equipment that's in the water. That's the way it goes. Good afternoon. Welcome to the Thompson, a research vessel operated by the university and supported by NSF. We have a show for you this afternoon. It's not exactly what we had in mind, but as some of the earlier footage showed you, a storm has come up and we have coped with it in the best way we can. It's very, very difficult when you compare the kind of weather that we're subjected to right now to what we were dealing with only 24 hours ago. Today we have a very serious problem, which you can see in the background, which is yesterday's effort with Jason, which was operating fine on the seafloor. Today we have a problem, which is the weather. So what you're seeing in front of you now is video that has been taken of structure on the seafloor, and that sulfide structure is called Hulk. It was originally named Incredible Hulk, and it was named by one of my colleagues back in the 80s who helped discover this field, and she now works at the National Science Foundation. The situation that we're in terms of the research that we have been conducting and the phenomenal high definition video that you will soon see, you are seeing now and you will soon see a good deal more of, is the result of what we call the Visions 05 cruise. The Visions 05 cruise is exemplary of a transition or a transformation that's taking place in the ocean sciences today. Let's take a look at a graphic that shows the upper portion of the ocean and all the complexity of all the processes that operate in the upper portion. And as the camera pans downward, we see that the ocean itself integrates the actions driven by the sun and the actions driven by the interior of the planet. This makes a very complicated system, and it's a very fundamental system. It's a system that the upper surface and the circulation of the water modulates the climate, and it takes a huge amount of energy and a lot of effort all the time, really, to understand the kinds of processes that operate in the upper water column. We've only lately discovered in the last 30 years that the bottom of the ocean is far more interesting than the vast wet desert we used to think it was. It turns out, as you'll see later in this program, that is a stunning array of life forms that are supported by what we call chemosynthesis, which is the opposite, or the counterpart, the chemical counterpart to photosynthesis, that is the process driven by sunlight. Now, the kinds of activities that we're studying on this particular program are shown in the lower part. It shows that there's a magma chamber there, and you can see where the plate dynamics, there's a cross-section through a plate, and then you can make the transition inward and inward and inward on that magma chamber, and pretty soon you see graphics of the sea floor with diagrammatically shown is a sulfide structure with black smoke coming out of the top of it. You'll see the black smoke, you'll see the sulfide structure, and that is another side of the ocean, poorly understood, difficult to access, and as you can see, very hard to stay focused on when the weather turns bad. Now, what we'd like also to do is look at the equipment that we have that we have to operate on. And what I'd like to do is I pass into the van, is pass by one of the heroes of the program, which is Jason. And Jason is the system that we've been using on the seafloor, and it's paired up on this particular cruise with a system called ABE, the Autonomous Benthic Explorer. The Autonomous Benthic Explorer is actually out in the ocean on the seafloor now as we speak, mapping. We had to pull Jason out because we operate Jason from the surface, but ABE operates on its own, no tether, no connection. We send it down to the seafloor, and it's operated by folks like Dana Yerger at Woods Hole Oceanographic Institution, and he is hovering over his computer now, watching ABE as it goes to sleep on the seafloor. So let's take a walk here into the van, and I'll be joining some of my colleagues in the van. Hi, that was a little tricky getting in here. We're really moving around a lot because the weather is actually, although the worst part of it is constrained to the outside, thank goodness, it sets us in motion with accelerations in unexpected directions on a regular basis. I'm sitting here next to my good friend and close colleague, Deborah Kelly, who we've been working together for almost 25 years, studying seafloor systems and submarine hydrothermal systems for quite a long time. Deb has quite a story to tell. And on my right is Peter Gerges, the assistant associate professor at Harvard, recently took the job at Harvard and has been working with us for the last 3 or 4 years. He and Deb have a phenomenally interesting program that they're working together. So what I'd like to do then is turn the discussion over to Deb to open with a discussion or a description of some of the footage you're about to see on the seafloor using this incredible high-definition camera that we have recently acquired from the company Insight Pacific. They delivered it with incredible speed, and it is an exquisite piece of work. Deb?
Sure, thanks, John. So yesterday, one of the big projects that we're working on right now is a project funded by the National Science Foundation. And the question that we're examining is what the upper temperature limit of life is. What are the most extreme environments? What are the most extreme conditions that life can live on the planet? And to do that, we can grow microorganisms in the lab, but it really doesn't give us information about what's really going on the seafloor in the environment that the microbes are experiencing. And so for this project, what we've done is develop an in situ microbial incubator. It's this instrument where we actually drill inside the walls of chimneys and put these instruments in that have chemical and thermal sensors. And so over a year or so, the organisms grow inside these instruments, and then we pull them out. So yesterday we explored a small chimney called Gremlin that we started working at for this cruise. It's a remarkable, nice little structure. It's not very large, maybe 4 or 5 meters high, and about the same across. And so it's covered with very dense communities of organisms, tube worms, limpets, variety of snails. And at the beginning of the cruise, we drilled, used the robotic vehicle Jason, which is pretty remarkable considering we're at 7,000 feet beneath the surface of the ocean. We drilled into the structure and placed one of these microbial incubators inside the walls. The hope is that over the duration of the cruise that the microbes have actually colonized this incubator. So what we did is yesterday actually we recovered one of these incubators, and what we see is that the incubators are very full of minerals and as well as microbial material, which we'll be looking at in terms of their genetics and culturing them. One of the interesting things that's come out of this work is the upper temperature limit of life that we know about is actually from a sulfide structure that we looked at in 1998, and that those organisms grow at 121 degrees C. Other work that we've done shows that the organisms probably exist at much higher temperatures, but the problem is it's very difficult to get access to the environment that those microbes are actually experiencing. So one of the reasons that we put Gremlin is that we really want to look at the conditions between the diffuse flow, where you see all those animals that are covering the structure, the outside organisms as well as those that grow inside. So I think, Pete, you might want to talk a little bit about what we saw yesterday at this structure.
One of the most interesting features about these hydrothermal vents is that whenever you look at a structure like that, it is more than just a pile of rocks. It's actually an amalgam of
Pile of rocks.
Pile of rocks. It's I'm a biologist. That's my bias. It's an amalgam of the minerals that have precipitated and the microbes that live there, and also the macrofauna that you see, the tube worms and a lot of the other critters, the mussels and limpets. That we find in some of these environments help consolidate this into a structure. So it's an environment where we can't work and just think about, say, just the biology or just the geology. And this is a perfect environment to conduct interdisciplinary studies that allow us to look at how microbes interact with the minerals and how they support the macrofauna that we see. So it's really exciting, and Gremlin is a great spot because over a very small area, we see a huge variety of environments, from the diffuse flow sites all the way to the high temperature flows. And that allows us to really look at just how far life can go, just the limits of life in a small spatial area. So.
Yeah, one of the things about Gremlin that we saw is, and one of the reasons we chose it, is it really, one of the interesting things about these environments, but also makes them difficult, is there's a lot of little microcosms, a lot of little different environments over the space of a meter or so. And so what Gremlin offers is an area where we have a black smoker chimney that's venting quite vigorously at over 300 degrees Celsius, so about 700 degrees Fahrenheit. And we also have this mound. If you were to look inside the mound, you can't even tell that there's rocks underneath it because the animals have covered them up. The good part they've covered. And in the background, what we see is an area of extinct chimneys. And one of the things that we're starting to recognize is that the microbial communities actually interact with the rocks themselves, and they can help in the oxidation processes. So there's this very interesting linkage between the microbes and the rocks. And in some cases, the microbial communities actually help precipitate out the minerals. So the Gremlin structure is kind of a small microcosm for some of the larger structures that we're going to be looking at later on this cruise and that we looked at yesterday, that in this one field get up to about 30 meters tall, so about 90 feet, and about 40 meters across. So this small structure is a great isolated place where we can look at processes.
That's right, and there's something else to keep in mind, and that is the organisms you see growing, or what we call chemosynthesis. That is that the microbes that grow in these environments, they do what plants do in our biosphere, in our terrestrial environments. They take carbon dioxide or inorganic carbon, and they make sugars, and they grow, and then grazers like cows and goats and things like that eat that, and then of course we eat the cows. Well, in these environments, well, some of us eat cows. In these environments, you have these microbes that take chemical energy and they do the exact same thing. They take inorganic to form organic, and that is the basis of the food chain in these environments. So this is all we call it a chemosynthetic community, meaning it's chemical dependence.
One of the interesting things that I've recently experienced just this morning was I came down about 3:30, 4 o'clock. Debbie Kelly, my dear friend, had been awake for almost 30 hours. She was almost dead with fatigue. She had just finished transferring one of the experiments that she had been working on, that she and Peter have been working on, and she went off to bed, and it was in the following 2 hours that basically the storm came up, and the experiment that is called the incubator, the microbial incubator insert, which you've seen pictures of in Gremlin, was left on an elevator, a device that has weights and flotation packages, and if we remove the weights acoustically, we can have it return to the surface virtually like an elevator. We just dial in floor 4000, and it comes all the way up to the surface. And at that point, that material is transferred into the laboratory, and there is almost an intellectual feeding frenzy as Peter and Min and the grad students and Debbie are sort of poring over this incredible artifact by design. We've pulled it out of a black smoker and brought it back up here, and the animals, the microbes anyway, that are living in it, are accessible, and it's exciting. But in the moment after Debbie went to bed thinking, I finally finished, everything's done. Peter was getting a little bit of shut-eye, expecting that he would be up working on it at dawn, and everything went to a faraway place in a handbasket. and we're still in the handbasket. So what I'd like to do is shift from that beautifully focused research program that these two colleagues are working on to a somewhat broader perspective, and we have the option of showing you the graphics that show a planetary scale location, a view of the planet with the water stripped out of the oceans, and in fact what you're seeing is, as a color indication, the age of the seafloor, generally sketched. And the age of the seafloor basically is youngest along the warm colors, and it ages away from these ridges that run down the middle of the ocean basins. Those ridges are places where the seafloor pulls apart at about the speed your fingernails grow, and it's the longest single mountain chain on the planet. It's more than 7,000 kilometers long. So what you have to understand is that entire feature, I made a mistake, it's 70,000 kilometers long. That feature is characterized by the Gremlin environment that Debbie showed you. And so at a planetary scale, the processes we're studying in detail on the seafloor are generalizable to this entire system. Now, if we move to the next slide and you see the ocean, you also see the plates. The plates are pulling apart, and that's where the young seafloor is. That's also where the hot magma is, and that's also where the submarine hydrothermal systems feed microbial life through chemosynthesis. The interesting point to be made is that there is a convenient plate just off the coast of Washington and Oregon called the Juan de Fuca plate. Now, what the Keck Foundation, the W.M. Keck Foundation did was fund our group to study the links between plate tectonic processes in general, that is earthquakes and volcanic eruptions, and the movement of fluid driven either by thermal transport or by actually physically being moved as a result of the earthquake activity. So the questions that we're facing now are ones we couldn't have even asked 10 or 15 years ago. What's the magnitude of this process on a global scale? Is it trivial? Is it significant? Is it comparable to the productivity that we see in the upper ocean in the form of phytoplankton growth? We don't actually know the answer. In fact, no one knows the answer because no one has even made yet a basic experiment that allows us to guess what the numbers are. That's what we're doing out here during this experiment. And I want to show you, if I can come in closer, on the ridge crest, which is actually where we are. Now there you can see on your screen Mothra, the Main Endeavor, High Rise, Solly Dog, and Sasquatch, and the seismometers are in fact distributed around them. They're the little yellow squares, and the red square is a special seismometer. You see on your graphics now that one of the seismometers on the seafloor and one on the ship. These are seismometers that are now in place. We've been placing them during the last couple weeks. What I want to show you is a fairly stunning expression of the earthquake activity that we've been measuring. This next graphic actually shows you 1 year's worth of earthquakes, and you'll see soon in a graphical portrayal an animation of those earthquakes. It's as if we're looking below the seafloor. The vertical columns that you're looking at are in fact the columns that connect to the seismometers on the surface. The surface is at the top of your screen, and we're rotating using the animation, and the individual earthquakes are colored as a function of the time of the year. The blues are early in the year and will move around. As you watch us moving around, you can actually see as the earthquakes exploding, and that's the time that they occur. So with this kind of constraint on the distribution of earthquakes through time and space, we have a program that also samples the water as it comes out of the seafloor. The water samples are very difficult to take, and it's important to realize that about 35 different systems have to be working perfectly for Jason and the arm on Jason to pick up the bottle and place it into a small black smoker orifice, which is basically the size of your fist. So the effort to take the sample of water also gives us the microbiology. The microbiology is what Peter studies. Our colleagues Marv Lilly and Dave Butterfield actually study the chemistry and the volcanic gases that come out of the seafloor in these waters. So this gives you a sense of how we are going about a totally new kind of science with Keck support to look at microbial productivity from below the seafloor, driven by processes of plate tectonics. Now, I'd like to introduce several people that are back in the studio. The first of them is Veronique Robigoux. Veronique was here on board 30 hours ago. She transferred in a late night transfer, the same transfer that brought Peter on board, and was back and flew into Seattle this morning. Veronique will be talking to you later about a program that she's involved in, which is operating on this cruise. The second person I'd like to introduce is Dr. Mark Emmert, the president of the University of Washington, and a new arrival at Washington approximately a year ago. Within the first month of his arrival, Mark came to sea with us for a day and a half on the Thompson as we steamed from Portland to Victoria. Mark, I wanted to open the possibility that you might have some either comments or a question or two for Deb or myself. It's an opportunity now to give us a hard time if you like.
Well, John, first of all, I'm sorry that the weather's not cooperating for you. You gave me a really nice day and a half on the Thompson, but I don't think the seas look quite like they're looking out there, and I'm sorry you're not able to get the live broadcast that we'd anticipated. But I have to tell you, sitting back here in Seattle in the studio, getting to see the quality of the high definition images that you're broadcasting to us, it's really quite exciting, and the educational prospects of using that technology is pretty self-evident. But tell me a little bit, and this can be Deb or Pete or you, what are the research applications for having those high-quality images coming out of Jason or any other device that you tend to use it with? How does this enable you to do your science better, and of course for our students to be partners in that process?
Hi, Mark. Thanks for joining us this morning or this afternoon. I've never really seen a lot of high definition video before, and it was very remarkable to see the imagery, the first images coming across the screen in high def. It was almost like you forgot that you were immersed in a sea of water down there, And these images, they really give us insights. And when you look, you can zoom in to fill up the screen of organisms that's the size of a quarter. And what we're starting to look at is it gives us these really beautiful images of the animal communities that grow on these structures. We can get a lot more information about how the fluids are forming these black smoker chimneys. We saw yesterday that a chimney that we had cut open in the morning over a period of about 8 hours grew about 1.5 feet. And so they really give us these remarkable views of the sea floor that we haven't seen before. One of the things that we used on this cruise was a light stand that you can think of as an underwater flashlight almost. When we use a robotic vehicle or are sitting in a submersible like Alvin, we get a very small image of the sea floor. And when we have this high definition imagery combined with these underwater flashlights, what we saw yesterday was these incredibly huge sulfide edifices with up to 20-30 black smokers on them, and we were able to see a much larger portion of the sea floor than we had previously. So the combination of different new technologies as well as this high-definition imagery is really giving us views of the sea floor that we've never seen before.
This is a new and a breaking and powerful technology that gives us the visual acuity that we might have on land. This is difficult to do in the deep sea because ocean water does not translate very effectively, doesn't transmit light very effectively. You can see maybe 20 meters at the most, and then the light is absorbed. So what we would like to try to do is come into the seafloor with these powerful new techniques, some of them designed for much more subtle light variations than most cameras can do, and many of them as sharply etched as you see them. The beauty of what can be done with this is to put it into stereo, mount the stereo systems on pillars, posts, or mobile units. And those mobile units, whether they're floating or they're on wheels like a big tractor, can move around on the seafloor, giving scientists the opportunity to actually move among the processes and the systems that you have already seen and you'll see more of, take samples, and with the kind of technologies we'll talk about next, we will be able to transmit this incredible bandwidth all the way back to the sea, from the seafloor to land, so that the robotic systems that are so important to us can actually be used. So that's the short answer, Mark.
I think also one of the things that we're starting to notice is that the hope is with these really amazing images that we can, not very many people get to go to the sea floor, and we try and bring lots of students out on our cruises, but there's still limited berthing. And so the hope is that through these really amazing imagery that we can show environments that are very important to how our planet operates that very, very few people get to see. And so it'll be a very interesting exchange between students and when we're out here or in the labs, when we can bring these imagery in and really show them places that they've never seen before.
So what I'd like to do is, Mark, is that an effective answer to your question?
Yeah, sure. I think that's a great answer. I was hoping Pete would still be there so I could tell him that I thought Deb's piles of rocks looked really terrific on HDTV.
We have a segment a little later in the program where Veronique will actually introduce the students and we'll hear some questions from them. The oceanography is a field in transition, in transformation. And that concept is best brought forward within a program called Orion. Orion is the overarching group that is overseeing the development of the Ocean Observatory Initiative. And we're very, very excited about the potentials of the Ocean Observatory Initiative, which is going to provide the support, we hope, to develop programs that will be absolutely revolutionary in our field. We will go to sea, of course. We will have complex experiments that we will conduct, but we will have a distributed global network of permanent and relocatable buoys that will be distributed over the entire planet. These will be in communication via satellite. In addition to the global arrays, we will have enhanced coastal capabilities that will, I'm showing you the mooring that will be an example of the mooring, and then we will have enhanced coastal networks that will already, many of them are in place, and they'll be expanded with this new technology. An additional component to the Orion program is called a regional cabled observatory, and the example is sketched in the graphic in front of you now, and that graphic shows a notional distribution of fiber optic cable, and the red dots are nodes where hundreds of instruments could be plugged in and distributed through the water column, below the seafloor, and on the seafloor to continually monitor change, to allow us to interact with change, and to be able to move forward in an interesting path scientifically. One of my favorite components or versions, I should say flavors, of this regional cable observatory concept is called Neptune, and our Canadian colleagues at the University of Victoria have begun to put Neptune in the water, and there is a partnership between the Canadians and the National Science Foundation to move down a path similar to that. And in simple terms, the components of Neptune will involve a tectonic plate, the Juan de Fuca plate, the handy pocket-sized one I mentioned earlier. It'll involve about 2,000 miles of fiber optic cable distributed on the seafloor as you see in your graphic. It'll have a network of submarine laboratories which are set up to do a wide variety of experiments interactively, so that the scientists on land, even students and teachers on land, can begin to manipulate some of the sensors and some of the robots. It'll involve the internet connected to the seafloor, so it's distributed across the world. The information goes ashore and is stored in places that have the capacity to accumulate petabytes per day of data. It'll have mobile platforms, many of which we now have and many of which are being developed. An example of this is the AID platform that's actually working for us right now. And it's designed to have about a 30-year life and to be extendable and relocatable. Now, how does that relate to what we're doing here? Partly the Keck Foundation funded us as a prelude to an experiment that might be done on a Neptune-like system. Partly, the Neptune-like system is going to empower us to study hydrothermal systems in ways we never imagined. But that's only, as you saw in that opening graphic, that's only a small fraction of the kinds of things that we want to be able to do in the ocean. So it looks as if we're looking at the Hulk video, and we shot this last night. We worked together to try to get this together. And as I understand, the place we are in that video is approaching the large structure. Can you take it from there?
Sure, John. So yesterday, one of the things I talked about was this underwater flashlight, which Dan Fenari and Deep Sea Power and Light put together for us. And this it really gives us this amazing view of the seafloor that we haven't had before. Hulk is a huge structure. It's been active probably for several thousand years, we think. And what it shows is these structures that are made out of rocks. You can think of them sort of like a big sponge or pumice, where we have high temperature hydrothermal fluids really just diffusing everywhere. And every place, because they're filled with the fluids have nutrients and gases that the animals use as their energy source. There's this incredibly luxuriant, vibrant community of animals that live on these structures. These tube worms that have these kind of almost like white plastic tubes with these beautiful red gills thrive in these environments. And that's one of the main things that Pete has studied for his PhD, and is now looking at the associations between the animal communities and the microbes. And I think what we're looking at is these close-up views of Hulk right now and some of the life that lives there.
This is the structure. We just passed a huge light tower which we placed on the sea floor, and we're now moving up the structure. The Jason vehicle, driven by the Jason pilots, are moving up the structure, and we're slowly approaching this upper reach that Debbie was just describing, and that will show an overhanging ledge, and you'll soon see a batch of tube worms that Peter will talk lovingly and passionately about, and we will move across, shortly we'll move across that scene.
The important point is to remember that this is an environment where we have really complex interactions between the geology and the biology. And so think of them as this big sponge that Deb was telling you about, as being a sponge in which microbes live, not unlike your kitchen sponge, but we'll talk about that later. So this is this sponge-like structure gives the microbes a chance to find the chemicals that they need from within the vent fluid and from within the seawater. And another thing is that in these diffuse flow sites, which you're probably looking at right now with a lot of these tube worms, the water temperatures there aren't as hot as what we see in the middle of the chimneys. That's a really important point. The water coming out of the vents is, can be up to 300 degrees Celsius.
What the folks are seeing is an overhanging ledge with this black smoker, and it's called a flange, and we're just about to rise above the flange into the region that Peter's describing.
Yeah, exactly. And the reason you don't see animals on that flange with those black smokers is it's really just too hot for them to live. And as we ascend up this structure and you begin to see these areas with lots of organisms, that's where the hot fluid from the hydrothermal vents mixes with the cooler seawater. And the temperatures there are about what you'd find in a nice warm bath. And these organisms love it just as much as we do. The tube worms in particular form these large colonies that you're looking at right now. And the tube worms are really, really wild. When I started as a grad student, it really blew me away that these tube worms that you're looking at have no mouth and no guts and no digestive tract. They're really sealed off bags. But inside their bodies, they host symbiotic bacteria. And you may ask, well, what's the point of that? Well, the bacteria that they host inside their bodies are these chemosynthetic bacteria again. They take inorganic carbon and make organic carbon. They make sugars, and they feed themselves and the tube worms. So it's a symbiosis of microbe and animal. And interestingly, despite not having a mouth or a gut, they're some of the fastest growing animals that we see on this planet. So this gives you a sense of what kind of life or what kinds of organisms can be supported in an environment that's devoid of sunlight.
The close-up shots that you see on the video now are the tube worms that Peter's talking about. They obviously are very, very fat and very happy, and they have a lot to eat. Well, a lot to digest. Well, a lot to.
They're busy. And if you look closely, you might see little tiny bumps on the tubes, and those are limpets, which are grazers, and they're very similar to the limpets that you find on the seashore. They're going around like little miniature herbivores, little cows, grazing on the bacterial mats that grow on the tubes.
Seafloor cows, anyway.
I don't know. Well, I got this. It's like the third time I mentioned cows during this broadcast. But bear with me. They're grazing on the microbial mats that live on these tubes. So it's not just tube worms and microbes. You have tube worms, limpets, crabs. They're pretty complex communities, and each day we often find ourselves discovering new and interesting species. So it's thrilling to be here studying these ecosystems because you never know what you're going to come across. And that's really fascinating. One of the interesting things about these environments is that it may give us some clues about the origins of life on our planet and certainly on other planets. And that's something that we have to, we really have to seize the opportunity here and be very diligent in our studies. One of the interesting things about this particular field is that you were looking at Gremlin earlier, and now you're looking at Hulk. And that gives us a sense of the scale here, right, Deb? I mean, we go from very small features to very large ones very quickly.
Right, and we don't often get to see those kinds of views. And actually, it was a very pleasant thing to go see Hulk yesterday because Margaret Leinen from National Science Foundation actually was the first person to see this from the submersible Alvin. And so we got to see this structure that normally we see just a little pinpoint of. And so we had a great time wandering around the structure Hulk. Particularly the top of it, it looks like a candelabra almost. We see most of the black smokers, they're finger-like chimneys of fine-grained black sulfide that emit about a lot of smoke that you've probably been looking at recently. And again, they're putting out fluids that are almost 700 degrees Fahrenheit. And the tops of these chimneys, they're very complex with lots of environments, but they're actually also remarkably beautiful to look at. And so we had a great time wandering around the Hulk edifice yesterday. During the rest of the cruise, we've actually drilled some holes inside of Hulk to look at the microbial communities and the environment inside of it. And hopefully if this storm ever decides to give us a break, we'll be able to go back and take some more water samples and retrieve some of these samples in the incubators and look at the microbes coupling the genetic studies that Pete's been working on.
So we're going to be in and out of more of the seafloor imagery, but what I'd like to do is invite Veronique, who is back in the studio on the campus of the University of Washington, with a group of students and teachers that have been part of her REVEL program. And I'm simply going to let Veronique, who is as passionate about the REVEL program and teachers as Peter is about tube worm physiology. All I can say is, Veronique, if you were out here, you would love what you see. You might even say something that Peter would identify with, like, holy cow.
Thank you, John. Thank you for this incredible display at Hulk, you guys. It's just wonderful to see it again, even from the studio at the University of Washington. One of the things I do agree with Peter about, even though I'm a geologist, is that I never think of the seafloor anymore as just rocks, but as rocks and life mixed together. And it just makes the whole field a lot more interesting. Before I continue talking about the geology...
You've gone over to the dark side.
It's not the dark side, John. What I would like to talk about a little bit about now is that, as you mentioned, that oceanography is going through a transformation. What is part of that transformation with Neptune and with the cable observatories is that nowadays we're also mixing non-traditional education with oceanography. And I think the REVEL program that we've been running at the University of Washington has been an example of that in preparing the next generation of being able to be involved in such interdisciplinary, but also, we have to face it, really complex type of science that we're getting into nowadays. And to illustrate this, we've invited, and actually we're honored to have here a generation of students that's been involved in that kind of research through their teachers who have participated in the REVEL program. And you haven't mentioned yet, but you have 5 teachers on board working with you side by side and learning about all this incredible, or all the Incredibles experiment that you are doing right now. So just now, I'd like to introduce you to some of the students that are present on the studio today, and then give them a chance to ask their question directly to you, John, Deb, and Pete, while you're out there. I want to introduce first Ashley Rose, who is a senior at the Orca Academy, and the Orca Academy stands for Ocean Research College Academy, which is located in Everett in Washington. And then I want to introduce Nick. Nick is an 8th grader at Tolt Middle School in Tolt, Washington. Welcome, Nick. And Bethany, who's also a senior at the Orca Academy. And all of them have teachers that have been previous revelers. We call the teachers that have gone through this program revelers. And they represent a generation of students that have actually been involved in that kind of research one way or the other. And they certainly have shared the enthusiasm of their teachers for this environment. So I would like you to take the floor now and ask questions to John and Debbie about what you've seen, or some of the questions that have might arisen while you assisted to this program.
Well, this is a question for all of you. I was wondering, what kind of experiments are you going to do with the microbes that you've brought up from the incubator?
Sure, that's a great question. So these microorganisms, one of the interesting things, it's almost unlimited experiments you can imagine, because it's estimated that we only know about 1% of the kinds of microbial life that lives in the sea floor. So there's this vast ecosystem that we really know nothing about. So every cruise is a discovery for us. Every experiment we find new organisms. So one of the things that we'll be doing is the microbiologists, some of the students working on this program, and as well as Pete, what they do is they actually take pieces of the rock out of the incubators that we've recovered, and they put them in test tubes, and they give them different kinds of nutrients, because we don't even know enough yet to understand quite what all these organisms use for their nutrients. And so that we test out a variety of menus, so you should say, and grow these organisms at different kinds of temperatures. So one of the things we'll do is try and see if these organisms will grow at temperatures over 120 degrees Celsius. The other thing we'll do, which is really a fingerprint, it's the kind of work that Dee Dee and Ginger and Pete work on, is really looking at the genetic fingerprint of these organisms. one of the remarkable finds that's come out of the incubator project is that for reasons we don't understand, organisms called Coryarchaeota, which are genetically some of the oldest organisms that we know about on the planet, for some reason they like these very, very high energy environments in the mixing zones between the seawater and hydrothermal fluids, and it looks like these incubator experiments actually enrich for these organisms. So there's a whole diverse array of questions that we can look at, and we're going to be, it's probably several years worth of work now from the samples that we've got to understand what's going on with them. So I'll pass it back to you, Vera.
Thank you, Deb. Maybe we'll go to Nick next and see if Nick is curious about something that you're doing on the research vessel right now.
Yeah, this question is directed mainly to John, but all of you will probably be able to say something towards it. How much will it help with this stuff to be able to go to the other planets, like to Jupiter's moon Europa? How much will this help learn about those planets and their ecosystems?
Nick, I want to thank you for that question. That's a fantastic question, and it happens to be one of my favorite topics. So I will use more discipline than I'm used to using in answering. I will talk only for 4.5 hours about the topic. Nick, one of the things that we really believe is exciting about the research we're doing is that there are other planets, some of the moons of Jupiter, and certainly in other solar systems there will be comparable bodies where volcanism, which is a natural consequence of planetary accretion and radioactivity, must take place, and in many of them they go through core formation, which expels a lot of the volatiles to the surface, and that results in there being the potential for an ocean. The interplay between volcanism and oceans is liable to be a theme that's replicated throughout many of the planets that form throughout certainly our galaxy and beyond. So we're quite excited, Nick, about the potential of the research that we're doing, and we would invite people who ask questions like you do to come to grad school and come to sea with us, because that would be good for us, and I think it'd be good for you, unless you get seasick.
Okay, thank you, John. Thank you for this great answer. And actually, it doesn't really matter if you get seasick, because I get seasick and I go to sea all the time, and I survive.
That's true.
And I still do well. Another question from Bethany.
You have designer shades of green.
Another question from Bethany from the Orca Academy before we move on to another generation of scientists that's also present in the studio.
Hi, my question was, since the hydrothermal events are so big, how do you get like the full picture of the vents, and how long does that process take?
That's a really good question. One of the ways that we can look at these very large structures, again, it's a difficult challenge for us because we're normally looking at little snapshots on the seafloor. But the new technologies that we're starting to use, the overview of JSON, vehicles such as Hercules, like Bob Ballard uses, and JSON, they really give us an overview that we don't get to see very often. And so what we do is we take these underwater cameras, we'll use them, we can use the high definition camera that we're looking at right now, and we drive up and down across these structures, and basically it would be like mapping a building maybe with a helicopter, where you fly up the side of the skyscraper and take images as you go along. And then what we can do is we can actually mosaic or piece, it's like a jigsaw puzzle, we piece all these images back together, and we can get entire overviews of structures that are up to, say, 200 feet tall. It takes a very long time to do that, but we are getting better at it. These autonomous vehicles in the future may be able to do that too, where you can actually pre-program. We've done this with JSON before. You program it, it goes under computer control. It's a remarkable technology feat because you're actually using a satellite system that interacts with the ship interacts with the vehicle, and in some cases there's no people really involved. It's all done through remote control. Dana Yerger at Woods Hole has been great doing this with the vehicle JSON, and you can actually move up and down these structures in a very rigorous fashion, moving centimeters per second, and image the entire structures. And then we have a great jigsaw puzzle to put all the images back together. But it does give us views that we really don't get to see in any other way. So Vera, back to you.
Thank you, Deb. This was great, and this is, this is a kind of mapping that's really close to my heart, since this is what I love doing when I'm out there with you. I just want to say a few words about the REVEL project, just in terms of what we're trying to accomplish with the teachers that are then translating the kind of research that's being done while you're actually exploring the ocean, and then translate it into the classroom. What we are trying to accomplish is to really promote a change within the teachers, and also foster teachers that are going to be more comfortable with the scientific process as it's done currently, and so that they can actually translate that in the classroom, but more than translate that, actually collaborate with their students so that they can explore how to do science, instead of exploring how other people do science through books and other static resources. And one of the cornerstones of the research project is actually that teachers do research when they are on board with us, and we take up to 8 or 9 teachers every year through this program at sea, and 5 of them, I just left 5 of them behind yesterday when I left the research Thompson. Now I would like to introduce Chris Ludwig, who's also a student, and certainly the next generation of scientists that's coming, and that can see how they're going to be able to use the type of technology that are starting to develop within the Neptune framework and the Observatory framework. And Chris, I'm happy to have Chris besides me, because I met Chris when she was very, very young on another expedition on another hydrothermal vent system in the biomass basin, while we were working together with Bob Ballard in the early days of transmitting information from the sea to land. So Chris, I would like you to tell us how you actually envision using this in your career, since you are the next generation of scientists that's upcoming.
Yeah, thanks. I'd be happy to. I actually, as Veronique mentioned, had a chance to go on my first ocean research expedition when I was about the age as some of the students we have in our studio here today. I was 16. And that was the first time that we were transmitting live imagery from the sea floor. And obviously it stuck. I'm still doing the same work now as a graduate student at the University of Washington. And during my time being involved in vent research, I've seen leaps and bounds in the technology and in the science that we've been able to use. And I have to say, bottom line, it's an extremely exciting time to be a student and an entering young scientist into the field. I've been at the University of Washington for 4 years now, and have even seen developments towards designing the Neptune program. And it's really exciting to see not only the developments within oceanography, but seeing scientists who may not normally work together or team up together for programs come together to work and design the technology, the data management, and the implementation of some of these programs like the Neptune that only a few years ago was just a dream of many oceanographers and is slowly becoming a reality. And it's really a treat to be seeing that develop in something. It's a tool that I look forward to using as a scientist down the road. So, John, thanks for your visions, and I'll look forward to using Neptune.
We are still transitioning on the ship, and so in the meantime, maybe we can get another question from one of you on the floor.
I have another question if there's time.
Ashley, sure.
Specifically for the tube worms, if they don't have a mouth or a digestive system, what is the use of their plumes that seem to be waving about in the hydrothermal vents?
Again, that's a great question, and it's a question that has probably multiple answers. The tube worm plume is effectively like gills in a sense, and it actually is capable of picking up the water, cycling the water down into the trophosome, or that body cavity that Peter talked about. And in fact, the tube worm cycles the warm vent fluids that are nutrient rich down to the microbes. The reddish color, the brilliant red color, which kind of surprises us in the deep ocean where there isn't much light, is actually because it contains hemoglobin, which is very effective at transferring oxygen. So that's basically the part of the chemical process, the chemosynthesis that actually operates in the tube worm system, if you want to call it that, because the tube worm itself it depends entirely on the symbiotic microbes that live within its body cavity. Thanks for that great question. Vero, will you give us a blessing as we try to appease the gods of the ocean out here?
Absolutely. You have my blessing, John. And thank you very much for responding to all of our questions and our curiosity here in the studio. And we wish you good sailing for the next 6 or 7 days that you're going to be out there. We know the weather is going to get worse tomorrow, so we'll all be thinking about you and hoping that you get back in the water and continue to do your science very quickly. Thank you very much to all of you on board.
Let me simply say that we very much want the young folks that were with you and the rest of the young folks around the country in places like Dwight Englewood, in places in Canada that are watching. We want them to seriously consider oceanography because the opportunities are growing dramatically in the coming several decades. So it's a lot of fun, although it may look like it's a little complicated right now. A few closing comments I'd like to make. Again, I'd like to contrast what today is like versus what yesterday is like, and remind you in a sense that if you take a sort of a broader view, what the HD camera brings to us is a beautiful, exquisite refinement on the techniques that were early on developed by Bob Ballard, who is one of the great visionaries of our field. It was Bob that originally designed the first Jason. It was Bob and Dana Yerger and Andy Bowen and a number of the colleagues that are out here, Jonathan Howland, a number of the folks that are with us helping us. Actually, we're part of Bob's original team, and we're very thankful for that. We're standing on those shoulders for sure. At this point, I'd like to end it. I'd like to end it and say thank you very much for joining us. It's been a long and harrowing day, and we hope you've enjoyed what we've done. Thank you.
Visions 05, Expedition to the Underwater Volcanoes of the Northeast Pacific, is a joint project of the National Science Foundation, the W.M. Keck Foundation, Neptune, the University of Washington, the Pacific Northwest Gigapod, and the Research Channel. Special thanks to the Neptune Ocean Observatory project partners and participants. Visions 05 Expedition technical and equipment support provided by